All right, I guess we'll get started because we're at 4 minutes past. So to our slides. So welcome everybody to our Lattice Lightsheet User Group. And I'm Claire, so I'm part of one of the co-chairs of the group. If we want to go to the next slide, Alex. So today we'll start with a welcome. We'll go through the agenda, some announcements, and then we have Yushi presenting today. Is Wes? No, it's just Yushi. And then we'll have some time for question and answer. And we did an update for our next meeting. Our next meeting is not April 1st, but we can come to that at the end. So we'll also be recording the scientific talk so that we can share that on our YouTube page for people who might want to watch again or watch later. Next slide. So as I mentioned, I'm Claire Brown, and we also have Brandon Scott, who's co-chair of the group at South Dakota School of Mines and Technology. Nikki Biley is away this week, so she's not with us today, but she's the program coordinator for BINA. And I'd also like to introduce Alex Cotton, who has joined BINA as our program administrator, has a lot of experience in science administration, and is doing a lot of the behind the scenes work to keep everything in BINA moving forward. So welcome, Alex. So we were going to ask you to put your name, your facility, and if you'd like to put your facility or your institution and company, and maybe if you have a major challenge or a win that you've experienced lately, could be related to Lattice Lightsheet or not, in the chat. And we've also want to keep track of attendance in our running notes. So there's a link in the chat for the running notes if you want to go in and just put in your name and your institution or company. Next slide. So we have a few announcements of different BINA activities. So if you want to hear more from me, I'll be giving a career talk on Thursday in the Career Speaker Series. And then we have another talk on April 16th on BINA Insights and Educational Tools from Industry from one of our corporate partners, Amita Gorer from Zeiss. Just a reminder of all the user group meetings happening in April. So you can find this information on our website under activities page. So we have our Lattice Lightsheet group now. There's an expansion microscopy group meeting tomorrow. There is a career speaker series that's ongoing. We also have our automated image management and metadata annotation group talking about image metadata and how to collect that. And then we have our STED user group next week on April 10th. We also have our open microscopy environment meeting that we're running in collaboration with several partners, including HHMI Janelia, the Allen Institute, Jackson Labs, German Bioimaging, University of Zurich, and then the OME community. And that's coming up at the end of April in Woods Hole. And registration is still open. There's still a few spots. So if you're interested in participating in that community meeting, there's a QR code there as well as the web link. We have lots of things going on. So we also have the annual Community Congress coming up in October in Montreal that we're hosting. And this is going to be in combination with the Global Bioimaging Exchange of Experience meeting. The registration opens next week on April 7th. And we really hope to see some of you in Montreal. We will be having some talks about Lightsheet during the conference for sure. We also have a core facility management course the week before the Community Congress, also in Montreal. And we're receiving applications for that course now. So there's a QR code there. And there's also travel grants available from both BINA and Global Bioimaging. If you want to take a look there, you can apply as well. And continued thanks to our BNS sponsors who help support the network. So these are our platinum sponsors: AVR Optics, Evident, EBD, Dot Photon, Leica, and TomoCube. So thank you for your continued support of the network. And I think that might be our last slide, yes. So I'll turn it over to Brandon to introduce our speaker.
Thank you, Claire. So yeah, it's a great privilege to introduce everybody to Yu Shi. He was a postdoc in Wes Legan's lab at UNC. He's recently, while he was there, he's published a number of really insightful papers. I think the one he's going to talk about today most is this smart lattice light sheet microscopy approach that they developed there, which happened to be, the cover of Nature Methods. So I think it's a pretty awesome thing there. And he's recently started his own lab at Western University in Ontario. And I think he actually marks the first of our Canadian presenters, which is quite exciting. So you go ahead and take it away.
Yeah, thank you very much. So let me share my screen and is it showing? Awesome. Thank you very much for the introduction, Brandon. And it's quite an honor to be the first Canadian, first speaker from a Canadian university here. So today I would like to briefly tell you some of the work that I've been done in postdoc, where we combine lattice light sheet with machine learning to build this smart lattice light sheet microscopy, and I'll show how we use it to image rare and complex cellular events. So before I jump in, I would like to use this opportunity to advertise my new lab a little bit. So my lab at Western University, I just started last September, and our lab is in the physics department, and my main research focus is study cellular biophysics, and particularly I'm very interested in understanding the connection between micro and macroscopic behavior inside a cell, like how does the micromolecular interaction leads to the macrocellular phenomena that we observe. And our lab focuses on mainly two subcellular structure. One is the cytoskeleton, and the other is the cell nucleus. And in parallel, we're also very interested in using various advanced technology to in this research process. For example, some micropulse array, which we use to study the mechanics of the cell, and more importantly, lattice light sheet microscopy, which is a pretty advanced microscopy technique. And so today, I mainly want to show you how do we combine this lattice light sheet microscopy together with AI, and we build this machine learning driven microscopy that can study these biological samples on its own. So the reason, what motivates this research is, I'm pretty sure you guys might be familiar with this pyramid of frustration in microscopy. This is basically saying that for a microscope, you have many different aspects, from the signal to noise ratio, sample health, phototoxicity, temporal resolution, spatial resolution, field of view, etc. And for any given microscope, you cannot really excel in all of this aspect, meaning that no microscope is perfect, and in order to excel on one of these corners, you always comes at a cost and sacrificing some of its other performance. And this idea, unfortunately, also applies to the lattice light sheet microscopy. And here's an example. This is a wonderful image taken by Dr. Basic's group. So this is a pig kidney epithelial cell. And here is an image taken with a lattice light sheet that covers about 900 micron, about 1 millimeter square times 10 microns, so it's a volume. And the resolution is about 200 nanometer in lateral resolution, 400 nanometer in axial resolution, which is about as good as you can get for the diffraction limited microscopy technique. So in this image, they capture this, and if you zoom in this image, you can see it captures some of the events of the cell migration, cell mitosis, and specifically, if you look at close enough, it can very rarely capture the cases where the mitosis goes wrong. For example, in one of these cells that I'm pointing towards, where individual cells dividing instead of into 2 cells into 3 cells, and this phenomenon is called aneuploidy. So if we look at the statistics of this movies, you can see that the movie is composed of about 1,000 frames, and it's taken at about 90-second frame per second. Sorry, 90 seconds every frame or every volume per se. So this is actually not a really fast speed if you say want to image the dynamics inside cell mitosis, but already this entire data set is at 45 terabytes. So we can imagine if, say, I want to image even a larger field of view, like across the entire cover slip, or if I say I want to image faster, like instead of 90 seconds per frame, I want to image, say, every 3 seconds or 6 seconds per volume. So you can imagine the data size is just going to explode, and the amount of data you cannot really, it's going to be very challenging for you to handle. So what leads to this kind of problem or this kind of challenges that actually also apply to other advanced microscopy is that a lot of advanced microscopy are very hard to operate. And specifically in the case of the lattice lightsheet microscopy, this is because if you compare the single field of view from the lattice lightsheet microscopy versus what you get from the wide field, you can see that the image taken with the lattice lightsheet microscopy is a lot smaller field of view compared with the wide field. So this means that if you're trying to navigating just in the lattice lightsheet microscopy, find some target cell that you're interested in imaging, it's going to be a lot more time consuming. And this task is also going to be increasingly challenged if you say want to image some very rare events or some events that are highly transient. Speaking from my experience using the lattice lightsheet microscopy when I was a postdoc, actually a lot of the time the really rate limiting step for operating the microscope is really finding a sample to image rather than the imaging process itself. So altogether, and with the recent advance in AI and deep learning, this leads us to ask whether we can combine the two and build a microscope that can let the AI to drive the microscope automatically. So of course, the answers to this question is yes. Otherwise, there will not be this talk. And in particular, what we do is we build a customized microscope that is basically based on the customized lattice lightsheet microscopy back in Genelia, but there are some modification built on top of it. So rather than having the typical like 2 objective setup, we have 3 objectives. And in this way, we can operate the microscope in 2 different image modalities. On one hand, with the with the objective that underneath, we can use this inverted objective objective as a wide field microscopy. So this can allow us to do sample scanning. And on the other hand, with those 2 objectives that on top of it, which forms the lattice lightsheet setup, we can use it to do high resolution imaging after the target sample is found. And we also apply machine learning in this case, so that on one hand, it can accelerate the finding of the target, and on the other hand, this will allow us to automatically switch between the wide field microscopy mode and the lattice lightsheet microscopy mode, so that we can do all this in an automated fashion. As we know that there are a lot of machine learning networks that are currently available, So in terms of choosing the deep learning network to specifically for this task, we use a network called YOLO, which stands for you only look once. So this is open source. This is open source deep learning network. The advantage of it is that it's simultaneously optimized the bounding box for the object and its classification. So in case you're not familiar with the with machine learning and image classification, so in terms of finding the so what we're doing is actually a two-step path. So we need to, from the image, we need to identify where the cell is, and then we need to classify what kind of cell it is, whether it's of our interest or not. So the first step here is really to draw out boxes around the object, which are called the bounding box. Like in this image, you can see draw boxes around the person, around the backpack, around dog, and around the ski. And then the next step is you need to classify what is within this bounding box. And here is where you label like a person, whether it's a backpack, skis, or dogs. So the advantage of YOLO is that it's basically combine this drawing bounding box process, which is called image segmentation, and also identifying the content in the box, which is called classification. And because it's combined these two tasks together, rather than doing these two steps separately, it significantly increased the classification speed of identifying this the samples. And here I'm actually showing a movie of this YOLO network that's operating in real time. And as you can see, this can be operated in real time at video rate. And it's actually this is actually one of its applications, which is automated driving, where you can see is identifying all the objects in the camera, and also label what it is, whether it's a car, whether it's a person, or it's traffic lights. So in our case, we here show we apply this YOLO network into two separate scenarios. The first scenario is where we use it to detect cell mitosis, where the cells are cells in division. And the second case is where we use it to identify the process of immune cell formation. So I would like to introduce a little bit about the training, because the process of how we train the YOLO network specifically for the cell mitosis, because it's actually not really a trivial task. The reason for this is because when you think about cell mitosis, on the entire cover slip, only a very small proportion of the cells are really going through dividing. This means that most of the cells are going to be interfaced, and it's really a very small proportion of the cell that we're very interested in. So you can imagine if, say, I want to, because one of the things I need to do in order to train this deep learning network is to label where those cells are and what class or what mitotic stage the cells are in. So if I do this manually, then what's going to happen is most of the cells are going to be in the interface, which is the stage that I'm not very interested in. And very rarely I can run into cells that are actually in mitosis, which are the things that I'm interested in. So this one not only makes me need to go through a huge amount of cells, but also the labeling efficiency is going to be very low. And more importantly, the training classes is going to be very heavily imbalanced. Like maybe about 99% of the cells are going to be interfaced, but only 1% of the cells are going to be in different stages of the mitosis. So in order to overcome this challenge, what we do is we actually first adopt automated cropping and segmentation networks so that we can identify the location of the cells are, and we can draw those bounding boxes. Draw those bounding boxes near the cell. And in case you heard of this, we use the network called CellPost to do the cell segmentation. And then after this step, we then train customized convolutional neural network. And this network will not really identify where exactly what mitotic stages are the cells in, but rather it will just identify whether in the image all the cells are in interface, or there is any cell that is in the mitotic stages. And then based on this, we can sort of filter out a lot of the raw images that are not really of interest because it doesn't really carry any cells in the interface. Then we only segment those cells that contain at least one cell in mitosis, and then we can do the manual annotation and then to identify whether it's in detail whether the cells in prophase, metaphase, anaphase, or telophase. And then together, what we can do is we can use the raw input images, and we use the bounding boxes using the automated cell cropping and segmentation pipeline, and we use the classes that we do the manual annotation, and all together we use those three to train the YOLO network to locate and classify the cells in different mitotic stages. So by the end of the day, we can characterize the performance of our network using this thing called confusion matrix. So here the y-axis is basically the label or the classes of the cell that the YOLO network predicted, and then the x-axis is the label that the mitotic stage of the cell that it is truly in based on our annotation. And in the ideal world, you like to have the diagonal. And in each square is indicate the proportion of the cell that lies in each category. And in the ideal world, of course you want the diagonal to be 1 because you want the precision rate to be 100%. But in reality, unfortunately, this is not going to be the case. But as you can see, from our training, we can reach about 80% for the cell mitosis. And for the immune synapse formation, we can reach an accuracy of about 70%. And then the next thing that we do is we now have a YOLO network that can be used to detect cell. Then we need to combine it with the lattice lightsheet microscopy so that it automatically operates. And then what we do is we first use the inverted objective to do a wide field scanning. And so here we do a scan of 900 micrometers square, and it can be done within just 6 seconds. And then we pass this image into the YOLO network, and it will detect what mitotic stages are the cells in. For example, you can detect there's a telophase cell in this image. And what I want to say is that this detection process for this entire 1 millimeter square images is only taking about 1 second. So this means that it's approximately it's about processing about 200 cells every second, which is something that cannot really be achieved by human. And then on top of it, we can use it to generate we can use it to basically do real-time cell detection. And by the end of the day, we can get a list of cells in different mitotic stages that we're interested in. And then based on this list of coordinates, we can operate the microscope in 2 different modalities. The first one is we can have a fixed biological sample. And then in this case, rather than just scanning across a 1 millimeter square field of view, we can square over 100 millimeters square. And this takes about 15 minutes to scan. And this actually covers basically the entire area of a cover slip. And then based on that, we can pass it to the YOLO network to detect the cells that we're interested in. So in this case, we demonstrated with a cell division. And as I said, the cell division comprised only less than 1% of the total population. And it took about 10 minutes to find all of the target cell. Altogether, we have about 200 mitotic cells. And then we can use the lattice lightsheet microscopy to go to each position and then collect a high resolution 3D volume of the image. And then by the end of the day, we have this gallery of mitotic cells that are in different mitotic stages. So the next question, so now we have this amazing data sets. The next question is, what do we do with it? What do we do with it? So at the very minimum, what we can do is we can so at the very minimum, what we can do is that we can use it to use the list of coordinates to generate the generated by the deep learning network to do some populational statistics. So here, what we do is we study the number of cells in different mitotic stages under both the control and the Taxol treated condition. So Taxol here, Taxol is a cancer drug that's has a known effect of slow down the cell mitotic process. And as you can see, if we now just, for starters, let's just look at the control cells, you can see the majority of the mitotic cells are in the metaphase, which explains why a lot of the literature published when they look at the mitotic cell is in metaphase. But if you look at the prophase, which is actually the rarest class of the mitotic stages, you can see it only possesses less than 0.02%. And this means that out of 10,000 cells, you only have about like 1 or 2 cells that is in prophase. So you can imagine how painful it is if you ask like a postdoc or graduate students to find the cells on the cover slip manually. And another thing that we notice is that if you compare the control condition versus the Taxol treated condition, you can see that there's a significantly less number of cells that proportion of the cells that enters into the anaphase or telophase after being treated with a Taxol. And this actually indicates that the mitotic cells actually are stuck in the metaphase under the Taxol treatment. So on top of these populational statistics, we can also capture the subcellular structure of the cells because we can also do the lattice lightsheet scanning at each location of the mitotic cells. And for example, here we show the detailed configurations of the of images with higher 3D resolution of the kinetochores and the which is in magenta, chromosomes, which is in cyan, and then microtubule, which is in yellow. And what I want to mention is that using this smart lattice mesh microscopy, we can collect hundreds of this kind of volumetric images, which give us a pretty strong statistical power so that we can do some quantitative characterization of subcellular structures during mitosis. So the first thing that we can look at is we can look at this structure called kinetochore, which is the binding site of the microtubule onto the chromosome. And what we can do, the first thing that we do is that we can look at the plane where most of the kinetochore resides in, and this is called the metaphase plane. And we identify this plate by doing a 3D fitting to figure out what's the localization of the kinetochores, and then we can use the principal component analysis to figure out the plane that most of the kinetochores resides in, and then we can study what is the relative orientation of this plane. So in this cartoon is the plane that is composed of the green axis and the red axis, and then we can study the relative orientation of this plane to the cover glass, so which is indicated by this angle theta. So then, if we plot out the distribution of this angle theta in both the prometaphase and in the metaphase, you can see that in the prometaphase, most of the cells, the metaphase plate orients almost in parallel to the cover glass, whereas in the metaphase, this distribution got heavily shifted towards about 90 degrees. This means that the metaphase plate from the prometaphase is actually more or less in parallel, whereas if you gradually enter the metaphase, it's going to be more or less perpendicular to the cover glass. And this process is called the metaphase plate reorientation. And very interestingly, what we found is that if we treat the cells with Taxol, we actually find that this metaphase plate reorientation is no longer there. As you can see that the distribution of the metaphase plate in the metaphase is actually now more or less uniform distributed rather than preferably distributed around 90 degrees. And then another thing that I want to highlight beyond just not just the metaphase plate was that if you look at the kinetochore or this those magenta dots in the prometaphase cells, you can see that it actually formed this kind of ring structure, and like you can see there is this inner circle that is basically void of any kinetochores. And because we have like hundreds of this kind of kinetochore images, then what we can do is that we can project all of those kinetochores onto the metaphase plates, and then we can average this and then we can average it across hundreds of cells. So by the end of the day, we can have this kind of almost like a heat map that indicate the probability of the kinetochores to appear within the metaphase plate. So again, if we look at this, look at this image, we can see that under the control condition for both the prometaphase and metaphase, you can see the circle, this magenta, which is the kinetochore, form this kind of ring structure, and we can further quantify it using the by looking at the intensity in the magenta channel, and then so here and then average radially across the cell. So here you can see for the from the solid line, which is the condition of which is the control condition, there is this clear peak. Sorry, there is this clear dip that is starting at 0 from the center. So, but very interestingly, when you treat the cell with the Taxol, you can see that this dip no longer exists, meaning that the ring structure of the metaphase this ring structure in prometaphase and metaphase no longer exists and is being disrupted by the Taxol. So previously, literature have been showing that this kind of ring structure actually facilitates the connection between the connection between microtubule to the chromosome, and what we show here is actually what the Taxol did is that beyond just stop the cells from entering the anaphase and metaphase. Anaphase and telophase. What it really does is actually disrupt the ring structure and also the metaphase plate orientation during the prometaphase and metaphase. So another structure that we can look at, not be away from the kinetochore, is the microtubule. So if we look at those images, you can see that for the microtubules, under the control condition, most of the cells have like two poles that's found in the microtubule, which is the spindle structure. And when we look at some of the cells in the treated with the 5 nanomole Taxol, you can see that under especially this images with the metaphase, you can see that there are actually 3 poles rather than just 2 poles in the spindle. And this kind of phenomena is actually, we can actually kind of quantify it and then look at the proportion of the cell that have multiple spindle structure inside the cell, that have multiple spindle structure. And as we see, for the control condition, we barely see any cell that have this kind of multiple spindle structure. But for the Taxol treated cell, there is a significantly a lot more cells that have shown this kind of multiple spindle structure. So this is kind of indicates that the taxol really actually change, not beyond just slow down the cell division. It really actually alters the mitotic spindles formation, and this is actually indication of some of the downstream process. For example, the spindle disruption in the spindle formation can also lead to some of the mitotic errors or genomic aberration in cells that are after finished dividing. So the next thing I want to mention is that beyond just the fixed cell, just those fixed samples, we can also image the cell in life. And in this mode, we'll have the microscope scanning again those 1 millimeter square areas, and then we can let it to detect whether there's a cell that's about to enter prophase, which is the beginning of the mitotic process. And this process can be repeated continuously, and then until a prophase cell is found, and then we can let the microscope to go to the lattice lightsheet microscopy mode, and then collect a 3D time lapse of this kind of cell mitosis process. So here here is actually a movie showing the kinetical structure and also the chromosome structure of the cells going through mitosis in life. And what's great about it is that all of this is being done in an automatic fashion. So you can imagine you can iteratively repeat this process, and then just set up the microscope script, and then let the microscope to run overnight. And then the next day, what you're going to have is you can have a gallery of this kind of live cell mitotic process. And here we're showing the kinetic dynamic across the mitotic process. And you can get this kind of gallery pretty easily, and also in an automated way. So the next thing, of course, is now we have this data set. What do we do with it? And the first thing we can do is we can localize the location of the kinetic cores, and we can study their trajectories. So we mentioned that in the metaphase plate reorientation, when we look at this fixed cell, but here in the live cell, we can again revisit it. And as we can see, indeed, when we localize all the kinetic core and study the metaphase plate orientation, we see that in the blue curve, which is the control cell, there is this clear trend of the reorientation of the metaphase plate as the angle changes. Whereas for the taxol treated condition, which is magenta line, you can see that the trend is much less significant, and there's actually barely a lot of reorientation process happens. So basically, this confirms our observation using in the fixed cell. And beyond that, we can also study the kinetical speed during different mitotic stages. And here we show the mitotic kinetical speed under the different condition, control and taxol treated condition, in both the prophase and also the prometaphase. And you can see there, what's very interesting is that there's not only just differences between the kinetical speed across different mitotic stages. For example, both in the control condition, there is a slight increase in the kinetical speed in the prometaphase compared with the prophase. But also, if you compare the kinetical speed between different conditions, you will notice that in the prophase, between the control and taxol treated condition, there is barely any differences. Whereas if I compare the prophase versus the prometaphase, in the prometaphase, there is actually a significant decrease in the kinetical speed in the prometaphase. So this actually indicates that the taxol affect the kinetical speed in actually stage specific mitotic stage specific manner. Another interesting that we can look at is to investigate the errors during the mitotic stages. So, for example, if I'm looking at this movie, which shows the dynamic of the kinetic cores, you can see that the kinetic cores occasionally become away from their bodies, like when it's indicated by red. But eventually, what happens is that you can go back into its bodies. So this is actually indicating that there's, even for the normal, even under the control condition where the cells goes through the mitotic process normally, there can still be mitotic error. There can still be mitotic errors happens, but the cell have a mechanism to correct them afterwards. And this kind of data are very hard to get using the fixed cell because you're really looking for this kind of very highly intrinsic moments throughout this dynamic process, whereas the fixed cell is just a random snapshot of this mitotic process. So we really need to use this to take these movies in order to study this dynamic, in order to study these processes. So luckily for us, we have tens or 20, we have like about 30 of such movies, and we can study this kind of mitotic errors in a quantitative manner. For example, we can look at and say how, like, what is the probability for this kind of mitotic errors happens when the kinetic core becomes an outlier. And as we see, is that during the prometaphase and anaphase, there is actually a higher probability of the kinetic core to become an outlier compared with the metaphase. But very interestingly, we can also look at how long does it take the cell to go from this mitotic errors to go and then correct it. And what we found is that actually in the prometaphase and anaphase, it actually took a lot faster for the cells to correct for this mitotic errors. Whereas for the metaphase, it took a lot longer for the errors to be corrected. And finally, I want to mention that although most of my talk focused on the mitotic, focus on cell mitosis as a benchmark for the smart lattice lightship microscopy, this idea can also be applied to other biological processes. For example, here we have cover glass that we co-culture target cell and T cells, and we can identify whether the T cell forms a immune synapses or it's just contacting with the cell, or there's no contaction at all. And again, we can go back and then collect a gallery of the cells forming this kind of immune synapses. And here the orange are for the T cell, the greens are for the target cell, and then the cyan are for the granulolysosome. So then based on this data, again, we can do some populational survey. So for example, here we look at samples with target cell having different antigen strengths. And we can use the smart lattice lightship microscopy to quantify the proportion of the T cell that form this kind of immune synapses under each condition. And in this figure, what we show is that there is this very mild increasing trend of the contact frequency between the immune and target cell as we gradually increase the antigen strength. And then another thing that we can do is, again, we can quantify some of the substrata structure. And in this case, we demonstrated with the granulolysosome. And here we can categorize the immune cell into different categories based on the distribution of the granulolysosome. To see whether it's located mostly near the interface between the T cell and the target cell, in the middle, or is further back from the interface between the T cell and target cell. And when we do that, what we end up finding is that as we increase the antigen strength, there is more likely for the granules to locate near the interface between the T cells and the target cell. So in summary, I want to we use the smart lattice lightship microscopy to offer an automated and efficient way of detecting rare events over a large field of view. We show that we can scan across areas at millimeter length scale and collect nanometer resolution images of target cells. And this data set can not only just provide us populational level statistics of those rare events, but also generate high spatial and temporal resolution 3D movies with statistically significant replicates. So in the end, I want to thank my postdoc supervisor, Dr. Wesley Legant, and all my fellow lab mates at Legant's lab. And also I want to thank all my collaborators that have been helped throughout this project. And the final thing I want to mention is that my lab is a pretty new lab. So if like any undergrads from your lab are interested in studying cellular biophysics using lattice lightshot microscopy, my lab is recruiting. So advertise those, please advertise those to your students, and I would really appreciate it. Thank you very much.
Wonderful. Thank you.
Thank you.
Does anyone have any questions?
Okay, we have a few coming through on the chat. Feel free if you'd like to ask it. You can come on camera if you'd like and ask, or I can read through the questions. The first one I can just read through here from Andy. What are fluorophores you use for inverted microscope and lattice light sheets, respectively?
So here we use, so for the mitotic cells, we it's not really I think for the kinetic core we use amnio green, and for the DNA or the chromosome we use M scarlet. And then use for inverted microscope. And so actually here we we do not really have two microscopes. It's the same. It's like the inverted mode. It's just a single microscope with two different modes. So the inverted microscope and lattice light sheet, they share the same fluorophores. And you have your sights on the next biological phenomena to quantify. So currently in our lab, we do not really have. I don't really have a specific task for this for machine learning. But back in our so I can speak for Dr. Legant. In our lab, we're also currently interested in applying this idea to neural cells, to study to neural cells, and to basically develop a high efficient way of identifying different cell types in the neural tissue. So basically increase the idea is still increase the throughput of the imaging pipeline. So do you imagine using the platform as a drug screening? That's something I've been seeing. I think can be one of the application. Although I'll be frank, I'm not a biologist, so that is something definitely beyond like out of my wheelhouse. But I do think another potential usage of this kind of imaging of this kind of technique is sort of a correlative imaging, because here we demonstrated on the lattice lightshot microscopy, but I don't really think like this technique is really specifically for the lattice Lightsheet microscopy. It's basically say you have you can say combine lattice Lightsheet microscopy, say maybe with even like EM or other thing where you use say like and I think there was a paper published in 2020 from Genelia, you basically combining the super resolution microscopy together with the cryo EM, which I think this can be something potentially of interest. For example, you can train network to say identify cells of interest using the super microscopy as the input, and then kind of identify the find this target the target list, and then feed those into the EM. So this can also increase the throughput in this way.
I have one question I would ask there. Is the software available? Is that open source or is that?
So we have the basically our own pipeline available. It should be in the SI of that Nature method paper. But the YOLO network is open source. Yeah. So we actually directly use YOLO, the YOLO network, which is an open source network.
Mm-hmm. And I assume this is using LabVIEW for controlling the light sheet?
Yeah, we use LabVIEW to control the light sheet. Yeah, but like all of the detection pipelines are actually done in Python because basically all you really need is just a list of coordinates, which is the output from the Python pipeline.
Yep, excellent.
Okay. Challenges using smart lattice Lightsheet. Yeah, like and thank you very much, Andy. So yeah, like alignment is always a tricky thing, and I would say specifically for the smart lattice for this task is like you want to, because like I mentioned, like for the YOLO network, for basically for any machine learning process, your precision rate is not going to be 100%. And one of the challenges is really how do you want to tune your network so that you can have the outputs mostly satisfy your needs. For example, if say I'm scanning like a fake cell, and in this case I want to identify as many mitotic cells as possible. So I would rather in this case I would say if it's detecting some cells in some like non-mitotic cells, not as a big deal, but I want to make my network as inclusive as possible. Whereas on the other hand, if I'm looking to a live cell process where each image is going to take me an hour to collect, and in this case, I want to make sure that I'm very, very confident with my detected cell. I want to make sure I'm like I want to make sure whatever cell the network is finding is going to be a mitotic cell. Otherwise, the cost is going to be huge. So this is basically, in one hand, you want the network to be as inclusive as possible, but in the other hand, you want the network to be as precise as possible. So this is one of the challenges that we had, and I would say like it's something that's kind of I would say task specific question. How feasible it is to implement a smart microscope solution for smart single molecule tracking. Oh, sorry. I think I didn't really explain this very well. So actually, the smart microscope is really only for the cell detection. It's not really for single molecule tracking. So for the single molecule tracking in this pipeline, we actually use basically the same analysis pipeline as the as that paper you mentioned in Nature Communication. So it's mostly based on the SMAP from Jonas group doing the molecular localization, and then we basically combine that with the 3D U-track to basically connect the trajectories. So for the single molecule tracking, there's not really any machine learning process that's involved in there.
Excellent. Well, thank you for a great presentation, and thank you everyone for the great questions their discussion. I think in the sake of time and respect for everybody, we should end the meeting now. But I think if you have more questions, I'm sure you would be happy to field any more questions via email to follow up with him. But thank you again for presenting for us, Yu. That was awesome.
Thank you very much. Thank you very much for having me.
Thank you, and thank you all for participating.
Thank you.